with the difference between what's in the syllabus and my announcements and what the WebEx thing says. So I'll just give people a few minutes. It gives me time to have my coffee too. Cat. All right, just a bit of feedback. Can you see the PowerPoint that I'm sharing with you?
Yeah, I can see it.
Awesome. Okay. I haven't used WebEx much before, so this is going to take a little bit of time for me to kind of get used to.
Okay. It has a lot of glitches every now and then.
Yeah, it's not my preferred choice, but we're basically told that's what we've got to use. So
Yeah.
that's what I'm going to use. So one thing I'm going to try and figure out is how to access the chat while we're in a shared thing. I'm not sure if I can. That's going to be a pain. Hang on, let's see. There we go. Where's that gone? Because that's another way that you can ask questions as we go through too, which may be useful. There it is, back again. Cat, really? I have to introduce Noelle to everybody else that's joining. She's probably going to be here for every lecture because she's a very loving cat and insists on getting love whenever I'm around, as you see. Say hello, Noelle. Unfortunately, that also means that my laptop and where I'm sitting is and me is completely covered in cat fur, which isn't always a good look. Absolutely terrible for allergies as well. All right, just a few more minutes. Sorry, everybody, there was a bit of a glitch with the WebEx scheduling. For some reason, it said 11:10. So I'm working on the assumption that everybody is going to be joining, at least who's going to join at least by then. So if you can just kind of twiddle your thumbs for another few minutes, and then we'll get started. Okay, so welcome to genetics summer semester. It's going to be a wild ride. There's no doubt about that. So, yeah, it's kind of crazy. I mean, genetics in a normal semester is challenging. It's got a lot of material, and this is basically at twice the speed. So, it'll be a very good test of your organizational skills, right? So essentially, the aim of this course really is to kind of in some ways build upon what you've learned in the intro bio sequence, particularly GenBio 1, right? This is really the kind of molecular and classical genetics part of GenBio 1, but kind of amped up. And also prepare you for the further courses that you're going to take, right? So uh, an obvious one is evolution, right? So genetics and evolution are really kind of two sides of the same coin. Evolution being really the kind of big scale picture of genetics over time. And what we're going to be doing this semester is really genetics in the kind of smaller scale stuff, right? From DNA structure up to some genomic stuff, right? So, uh, and really, a lot of courses that you'll be taking afterwards are going to lean fairly heavily on what you're learning this semester, right? So it's a really important foundational course. And to be honest, it's very much the one which will transition you, particularly if you're a more traditional student, you've come in just through high school, a little less so for non-traditional students. But it will help you transition from kind of the bulk learning type stuff that you're used to for kind of high school and the like to kind of the critical thinking that you need to be successful at university, right? And so that's really a big part of what this course is about. And so there's going to be a lot of content, right? But also a lot of Linda, can you do me a favor and mute your microphone, please? Thanks.
Sorry about that.
No worries. Uh, where did I go? Oh god, like the attention span of a goldfish right now. And also a lot of kind of big picture concepts, right? Because really genetics underlies everything biology. Even if you like, big fluffy pandas chewing on bamboo and stuff like that. And I personally hate giant pandas. They're my least favorite animal of all. Which if you talk to anybody about my favorite animals and least favorite animals, you'll hear that story. But even if you're interested in that kind of stuff, genetics underlies that, right? So genetics is really the kind of the foundation for pretty much all biology, whatever aspect of it that you're particularly interested in, right? Even if you're interested in like the big scale macrofauna type stuff. So it's going to be real tough, right? I'm going to be super honest with you ahead of time. This is a really challenging course to take over the summer. And the reason for that is that it's full on and we don't let up. There is no respite whatsoever. We don't even have 4th of July kind of giving us a break, which is a shame because, we're not teaching on the 4th of July. So really it's, we kind of start today and we don't let up until 28th of July, right? And it's going to be really intense, right? So there are some things you can do to help with that. Drink lots of coffee, obviously. And if you're not a coffee drinker, I really recommend it. I can see Angela is taking my advice. But organization is the key, right? So there are some things which are in this course and in the lab, which are designed to kind of help you, right? To help you build up your grade. And in the lecture, those are the homeworks. Pardon me. So if you look at the schedule, right, there's a syllabus and the schedule. The schedule has what we're doing on each day and also other important dates like exams and homeworks. So put all of those homeworks and exams in your calendar, right, on your iPhone or tablet or computer or whatever, right, and have a generous reminder, like at least a day ahead of time before the deadline, right, because those are easy money, right? You get a couple of chances at answering each question. None of them are, super difficult. It's a lot of language, right? So you can kind of build up your kind of dictionary of genetics terms, essentially, a lot of basic stuff that you need to know. That's 20% of your lecture grade, right? So that's a big chunk, right? If you do all of those, get them all right, essentially, you only need to get, was it 50 out of 80 percent to get a C or better, right? So that will get you a long way to where you need to be. But you need to stay on top of it, right? Because they're going to come up one after the other, right? There's no breathing space really. In the lab, and we'll talk about this a little bit later on, the weekly assignments, right, are the other way in which you can put away basically money towards your final grade. And really the lab as a whole, even though it's designed to kind of give you the skills that you need to be successful later on, is also there to help your course grade, right? So the lab is worth 25% of the course grade as a whole. And almost always people do a lot better in the lab than they do in the course, right? Because it's just stuff that, there aren't exams in the lab, right? It's not stuff you have to study for. It's things that you do every week, every lab, right? Which will accumulate towards your final grade, right? So keeping on top of that stuff is also really important. And if you do those two things together, that's what, God, I'd have to do the math. That's kind of painful. A reasonable chunk of your final course grade, right? And particularly because exams are hard, right? Exams are hard for everybody, and some people find them harder than others for reasons that aren't tied with their intellectual ability, right? So those things, the lab and the homeworks, are really there and designed to kind of help you build up your grade, and if necessary, hopefully not, but if necessary, compensate for difficulties that you might have in the exams, right? So don't waste those. They're really, really useful. And copy, more copy. Other than that, it's pretty simple, right? I'll talk about the lab more in lab when that is, 1:45, I think. But the rest of the lecture grade is exams, right? We're going to have 4 exams, pretty much one every 2 weeks, right? So it's kind of brutal, right? There's no getting around that. And the course is pretty much split up into 4 sections, right? So there'll be an exam for each one of those. There's, oh shoot, I'm not sure if I uploaded it. Make myself a note. Actually, let's put it, I'll get the process started here. That's where I wanted to go. All right, I'll leave that open so that I'll do that when we finish or when we take a break. There's also a file which I'm not just about to, but will today upload, which is essentially all of the course aims, right? And so you can use that as a checklist to basically compare what you know with what you're expected to know, right, in terms of the exams, right? So you can go through that and basically see, are there any things on here which I don't know? Because with the way that I do exams, every topic, every lecture is kind of equally represented by and large, right? As long as they're kind of equal in kind of content. And so you need to be able to kind of cover the whole deal, right? The exams are multiple choice. There are 40 questions per exam. They're going to be online, obviously, because we're not meeting in person. And They will be on Blackboard. Typically, I set them up so that they'll be started in the actual course period that they're scheduled for, and then I'll leave them open for the rest of the day until midnight of that day, right? So if any of you, and I'm saying this not just for people that are present, but also for the people that can't attend WebEx because they have other commitments, and I'm recording this so that those people can see this later, right? So some things may sound a little weird when I say it, but it's really there for those people as well. All right, so the exams will be open for the day of that exam, right, until midnight. Now you're going to have a time limit on how long you get to take it, right? And obviously that time limit will be a 1 class period, but you can take it at any time during that day. So if, for example, you don't get childcare or whatever during that particular period, or you've got to go doing a shift at last notice, then you can still take the exam. You're not going to be disadvantaged, right? You can just do it later. Okay, so that's the plan for the exams. Obviously, teaching online is going to be a challenge, and this is the first intentionally online course I've ever taught. Obviously, last semester was unintentional, right? I didn't plan on teaching genetics online last semester, but we ended up doing that nonetheless. And so essentially, I'm continuing what I did on there because that seemed to work pretty well by and large. The exam scores didn't really change much from when I've taught it before. So I think more or less I was providing the support materials and instruction that people needed. But basically, there's going to be three planks to this course, and this is in somewhat a repetition of what I've already said by announcement, but I'll say it again for the sake of clarity. The first are the PowerPoints, right? So all the PowerPoints for the whole course are uploaded in the course content section on Blackboard. They do have some notes in the, like if you kind of look at the bottom of each slide, let's see if I get back to this, right? So if you look down here, there's a notes section. I don't think I've got any notes in any of these. Let's see. Oh, here we go. So if you have the PowerPoint opened up and you don't have the notes showing, kind of pull that up. If you don't, it'll look kind of like that, right? So either click on notes or just kind of hover your cursor there and like pull it up, and there'll be extra information in there, right? So if there's a particularly nuggety concept that I want you to get, then there'll be extra information that will help you with that. Right, so all of the exams are based on the PowerPoints, right? So I don't ask you questions. I have to be careful with this. Certainly don't ask you any questions about information that's only in the textbook. Right, that's a definite. We can talk about the textbook in a little bit because there's been a bit of a issue about that recently. And maybe I'll ask you the odd question based on what we talk about in the WebEx sessions. But usually those are expansions of what we're talking about on the PowerPoints anyway, right? So if you are unable to attend these WebEx, or even if you're unable to watch them for whatever reason, you should still be fine, right? You're not going to be caught out by stuff which appears in the exam that you're like, what the hell? I didn't see any of that in the PowerPoints, and I studied them with a magnifying glass, right? So the aim is not to catch you out, right? The aim is to provide you the information you need, and you've got that already ready to go, right? There's nothing that's going to be, in terms of PowerPoints, uploaded or changed, right? It's already all there. You can read it all if you want to get ahead of the game. Okay, so that's one thing. The second is I've not finished them yet because stuff takes time, and I've had other stuff to do before starting this course. but you will have audio walkthroughs for all of these PowerPoints. And the aim is to, actually my aim is to finish all of those ideally by the beginning of the next week, depending on what other stuff that I've got to do work-wise and around the house. So that you have basically my dulcet tones walking you through these PowerPoints, right? So if I've got, let's say, this picture, and you can listen to this yourself, right? I mean, it's not a mystery. I'd be saying, well, basically describing what are the key points on this slide are, right? The key points are that it's anti-parallel double helix, right? We've got 4 different bases. You can see them here, joined by hydrogen bonds. We've got complementary base pairing, right? I'm going to walk you through that slide, and I'll do that for each individual PowerPoint. And I'm pretty good at it now at remembering, but I'll also tell you what slide I'm talking about in each PowerPoint. And the audio file has the same name as the PowerPoint I'm walking through, right? So you can kind of pair them together. So those should all be up very soon, and at the very least, they'll be up at least 2 days before your lecture to give you time to use them and go through them. Right. So what I'd recommend is, and this everybody has a different way of studying, but this would be a good way of doing it, is to go through these PowerPoints and listen to the audio walkthroughs at the same time. They're usually about 25, 30 minutes long, right? So it will give you a good heads up, essentially, about what we're going to talk about in the WebEx lectures, right? And I'd recommend doing that before we do this, because this is like the third part, this online kind of face-to-face, right? Sort of. It's like one face to lots of faces. I'd have to like scroll through all of you lot that are up here. It's kind of fun because I get to see like what your houses look like. Some people have pets. I had a student last semester who had a dog that snored. Sounded like Pete's dragon, basically, next to it, which was ever so cute. And the big point about this is it lets you ask questions, right? It lets you interact with me and kind of pick my brains and clarify things, which the PowerPoints and the walkthroughs alone don't, right? Because there's going to be a lot of information in these PowerPoints, and you might not understand some of them. Almost certainly you won't, actually, right? Because otherwise you would have already taken genetics, right? You already know this stuff. And so the main purpose of these WebEx things is for you to, stick your hand up. I think there's even a stick your hand up button, I think. I'm not 100% familiar with WebEx, to be honest. Oh yeah, there you go. Thank you, Alyssa. You stick your hand up, and presumably I get told that. Can actually someone do that so I can see what that looks like? Yeah. Oh, there you go. Not literally, Angela. I mean, that helps as well, obviously. But cool. Yeah, I got a little hands up from Gabrielle. Right. So you can do that. You can unmute your microphone and just say, hey, Professor Crook, you said such and such. Like, I don't get that. What do you mean? Or you can write it down in the chat box, because I've got the chat box kind of down here open up, and I'll be able to see any questions that you write there. You can even write them to me personally, right? Privately, if you don't want to see other people writing your, hello. So you don't, if you're worried about appearing stupid, you don't have to worry about appearing stupid. In fact, I'd really advise you not to worry about that. I really want you to ask questions. And so what I'll do in these sessions is basically go through the PowerPoints, but I'm going to be picking out like the key points that I really want you to study. I'm not going to just repeat what I've already done in the walkthroughs, the audio files, because you already have those, right? That's not really going to be super useful. What I want to do is basically go through these and then give you time to ask questions and pick my brains, and we can go back over different slides and things like that, right? And that's really, in the way in which you're going to really succeed, right? Because you can just kind of brain cram the material and listen to your walkthroughs and so on and so forth, and you'll probably do pretty well if you can do all of that. But if you can attend these WebEx sessions, or at the very least watch them, right, which will be a good enough second best, you'll also get the benefit of all those clarifications, right? What does it mean anti-parallel? Right, I know you know what that means. That's just a sorry, Karina, I can't let you change this stuff because then I'd have to re-upload it for everybody else to see. If you've got questions, though, please do feel free to ask, and I'll be more than happy to answer them for you. Right, so that's basically the strategy, and it's really up to all of you to figure out kind of what the best mix is. Oh, hey, no worries, Karina. The best strategy for you to study, right? But whatever you do, you gotta stay on top of your shit, because otherwise you'll be overwhelmed, right? Our first exam is, and you're probably not going to appreciate me telling you this because like no one does. Your first exam is next Thursday, right? So on the 18th, right? Your first homework is due this Friday, right? So this is just nuts, right? Hopefully, and this is like sincere hope of mine, you'll all power through this and you'll be like raring to go in the fall for whatever other classes you need to take then, right? But it's going to be a lot of work, and it's going to be, particularly if you have other commitments, either in terms of studying or family or work, it's going to require a lot of careful thought and organization, right? So, try and keep on top of your stuff. And if you have any problems with any of this, just send me an email. I know it's not ideal, like you don't have office hours, formally and everything. And if you can ask questions here during this, or if you don't want to ask those questions or talk about those concerns publicly, just send me an email and we can always arrange a private WebEx chat like it would be in office hours, right? So if you need the support is there for you. You just have to ask for it, okay? Right, so that's kind of the plan. And what I'll also do, and hopefully people will chime in, is I'll stop and ask questions, right? So does anybody have any questions? Also gives me time to drink my coffee. About any of the stuff that we talked about, the structure, the format, how we're going to interact. Again, if you do, you can either type them out, you can raise your hand, I guess, or you can just unmute your microphone and just ask. If there's more than one, then I'll just, separate you out in time. Any questions? Oh, okay. Homeworks. So homeworks are I can even do this fancy style. Let's stop. In theory, at least. Oh, share my web browser. That's a little bit worrying. Okay. Well, here are all my web tabs, which usually terrifies people for some reason. But Crabsticks. Let me go back. So yeah, I know. Part of that is my bloody house and car insurance. Got to sort out. So buggers keep increasing it every year for no apparent reason. Okay, so if you want to know where the homeworks are, and this is what I see, so hopefully it'll look similar to what you see. There is actually here we go, student preview. So this is supposed to be what you actually see. So exams and quizzes, right, they're all here. So you just got to click on them and then they start. So each one is due, pardon me, the day it says in the schedule, and it's due at midnight. So 11:59, you can start taking it up until that point. Now I do, and I'll probably cause myself and all a bunch of strife on that. If you do for any reason miss one, because you forget, it's normal, or you just had something come up that stopped you from doing it, or your internet crashed or whatever, you get one free pass a semester, right? So if you do forget or have an accident or whatever, you can't do it, and it's not an excused absence, it's just a sorry, professor, I forgot. Just send me an email and I'll open it up for you for 2 days, right? One per semester. Okay, so try not to use that if you don't have to. Okay, but it is there if you do need it. And so you just go into, actually, what does this actually look like when you go into it? There you go. This is fun. I can actually do this and see if I can answer the questions. You just go into it and then you begin the test, right? Obviously, I'm not going to do it because that will tell you what the answers are. But that's how you do the homeworks, right? So again, as you can see, they're just general questions on the material. They're really there to give you a hand in terms of learning the stuff, right? They're really a supplement. Yeah, that's fine. The course aims, when I get to uploading it, will be in this folder. The syllabus, which a whole bunch of you have found already, and the schedule are here. The readings for the textbooks are here. So the textbook is really supplementary information. So if it helps you to use a textbook, go buy it or rent it or borrow it from a friend, right? And here are some readings that will help you out, right? If you buy textbooks and then they sit on the shelf gathering dust and you're like, what the hell do I buy that for? Hang on, Olivia, I'll answer that in a second. Then don't bother buying it, right? So it's really up to you as to whether it suits your studying style to buy it or not. You're not obliged to. But if you do want to, here's some readings that may help, right? Syllabus, schedule, and then here are all the PowerPoints, right? So there are a couple missing because we're not going to cover them in a series that I have. So 14 and 19 are not there because, it's just extra stuff and I decided not to include it this time around. And here are all the audio walkthroughs, right? So these two are for the material we're going to cover today, and 3 to 11 will be stuck in there as soon as I've finished them, which will be over the next week, ideally. Okay, so that's pretty much all that there is in terms of content. Also, when you do your exams, they will appear here, right? So your online exams will appear in the exams and quizzes. Now, one thing that causes some unnecessary anxiety is that they won't appear until the beginning of the class period in which that exam is due to take place, right? So if you kind of come on here like 10 minutes beforehand, you're like, oh shit, the exam isn't here. Don't worry, it will magically appear at, what would it be, 11 a.m. of that day if it's in the first class period, okay? So let me get out of this. Let's see if I can. Don't share that, just so that you're. How do I stop sharing that? Okay, let's see if I can do share content instead. All right, just so you're not too traumatized by my excessive list of tabs. Okay, so Olivia, hopefully that answered your question. Both the PowerPoints, not the PowerPoints, the homeworks and the exams, the online exams will be in the same folder in the exams and quizzes folder on Blackboard. So hopefully that answers your question. Damn it, now where's my... Oh, awesome. Find out where my chat box has gone. Ah, crap it. Sometimes these things leave stuff to be desired. Where we go? Ah, there we go. Good stuff. Oh, I've even got a little picture of me down here too, so I can make sure you're not seeing just my bottom half of my head moving. Okay, any other questions?
I have a question.
Go for it. Who's that? Gabrielle?
Gabrielle, yes. For the homeworks, since there's like 10 questions per homework, are those questions based off of the PowerPoints and lectures or based off the text?
They're loosely associated with the PowerPoints and lectures, right? So they're not, they are based off of the PowerPoints and lectures, but they're really, I'd have to go back through and look at them, to be honest. They're not based on the textbook, that's for sure. But they're really just stuff to get you thinking about the material, right? So you kind of start processing like different aspects of the material, and also to give you the vocabulary that you need. Right? So those are the real kind of aims in that. They're always going to be associated with that particular topic or topics, right? So each homework is kind of reflects the lectures that you will have had before it, and maybe a little bit the lectures that you'll have after it. Again, I haven't looked at them for some time, but it's really just to kind of give you a little bit of revision and also give you a little bit of reinforcement of the material that we're studying. Does that make sense?
Yes, thank you so much for that.
Yeah, no worries. Did someone stick their hand up? Something appeared while I was looking at my
Oh, it was
chat.
probably me. I put my hand back down.
Oh, okay, cool. She is very, very cute and distracting. Yeah, talking about you. And whenever I give lectures online, she always just lies right here. There's a big pile of cat fur right next to me on the couch. All right, so if that's it, and oh, Gina, go for it.
Yes, can you hear me okay?
I can indeed.
Okay, great. I know you just sent an email saying that you were going to try to get a few copies of the textbook. I know you mentioned about them changing ownership or whatever. I guess my concern is because we're entitled to it supposedly, though. So that's why I was like, okay, well, can we get a copy?
Entitled it how in terms of the summer?
Included in the tuition.
Oh,
I think
okay.
starting a new program, so.
Right. I've got to apologize. This whole Jaguar Day One stuff is a complete mystery to the faculty.
Okay.
We have absolutely no idea what's going on, and the more we discover about it, the more we dislike it, to be honest.
Oh, no.
So I'll sort that out best as I can. I don't have a huge control about it. I typically just say, hey, this is the textbook we'll be using, and then they just get on with it. And for some reason, even though we've used the same textbook for the last 4 years, they didn't think that we were going to be using the textbook this semester. I don't know why. So there is actually, I've heard, like amongst the faculty grapevine, a way that you can opt out of that fee. So if you don't wish to use a textbook, or you've got a copy that a friend gave you, or whatever, you bought a cheap one off eBay, pardon me, you can actually opt out of that fee. I think it's like $75 via FAQ page somewhere. But again, I don't know. It was pretty much just dumped on us with very little notice, basically. So I know pretty much all that I knew about it beforehand was a never-ending supply of emails telling us about like some training that we had to do. Oh, okay. Hang on. Angela, do you mind if I share that with everybody? I'll just wait to. Okay, so Angela said that you should have received emails on how to opt out of the textbook. So maybe Angela, if you can send me a copy of one of those emails, and I'll be able to share that with the rest of the class in case they didn't get it or they've lost it. That would be really handy. But anyway, I'm working with the bookstore to get those books back in stock. And again, my apologies about that. The first I knew about it was when I checked my emails this morning and there's a whole bunch of stuff. I was like, oh crap, makes me look like an idiot basically. But anyway, so hopefully that will get sorted. And also, I've also heard, again, somewhat offhand, that the textbooks are free this summer. Yeah. Okay. Cool. Andrea, could you do me a favor and send me that email when you get a chance? I'll forward on the text of it to everybody else. Okay, I had another question from. Oops, got a little bit buried up here. Destiny. So watching the WebEx later, that's a good point. I forgot to talk about that. So I'm recording it right now, fingers crossed. First time with WebEx. And when the lecture's finished, and this will go for the lab as well, I will upload that to YouTube, and then I need to send you the playlist or the channel or whatever, and then you can watch that later at your leisure or leisure. So those I won't be doing it on Blackboard because Blackboard has like teeny tiny amount of storage space, and it will run out before the end of the semester. But those will be going up on YouTube, and you'll be able to watch those. Actually, you could even watch genetics from last semester. Wouldn't that be fun? And my astrobiology course too, in case you're like really bored and don't have anything to do. Cool. Okay, so any other questions? Anyway, as for the textbook stuff, as I find stuff out, I'll let you all know. But really, you probably know more about this Jaguar Day 1 thing than I do, to be honest. Oh, cool. Cool beans. Yay! I love questions. Questions are awesome. Otherwise, it feels like I'm just talking to my cat. That'd be super cute, wouldn't it, kitty cat? I'm not going to do any more of that. Don't worry. Just in case you're like, oh, baby talk to cats. Okay, so let's start on. These things shouldn't take too long today because these are pretty basic kind of introductions, right? It's kind of like ease you into the process so you get a feel for how we're going to do stuff and also give you some of the language that you need, right? So really these are just intro PowerPoints. The first two are by and large. The second one is a little bit more so, less so rather, but the first one definitely is. And so really like one of the cool things to get about this, and this is perspective that you might not necessarily have, right, is that genetics is actually a fairly new science, right? It's really started with Mendel, right? So he was the essentially the grandfather of modern genetics, right, with his work on peas and stuff in his monastery. Obviously, he didn't have a good book to read at the time. But anyway, he was really the first one that showed that kind of traits, right? And by trait, we mean a particular thing, right? So hair color, right? That's a trait. Love of cats, maybe? I don't know. But those traits kind of define the thing that you're talking about, right? So propensity to obesity, right, is a trait, right? But within that, everybody has different phenotypes based on their individual genotype and, their kind of environmental interactions, right? We're not going to talk a huge amount about gene by environment interactions, but we'll talk about it a little bit later on in the course. So anyway, phenotypes are really what version of that trait you express, right? So in terms of hair color, right? I'm like brown haired, like most people. But you have also obviously lots of different types of phenotypes. Red hair, for example, right, is a particular phenotype, but also that's based on your genotype, right? So genotype is really the collection of genetic information that is expressed in that phenotype, right? In the case of red hair, you probably have a mutation in a gene called MC1A, MC1R, there you go, which is a melanocortin receptor, right? If you don't have it, you don't produce melanin, so you express the underlying color of that melanin, which is kind of a reddish color. So Mendel was really the first person that showed that those traits are discrete, right? And that they're due to the inheritance of discrete bits of information. And that discrete word is really important because it means that essentially if you have two individuals with different phenotypes and you mate them together, you don't get an average, right, of those two. It's not a blending of that kind of those two different phenotypes, right? And that was very much what people thought up until Mendel. So, and obviously the one example often given is height, right? But as you know, you can have like both my parents, like tiny, like 5 foot. Actually, I wouldn't say tiny, like 5'6", 5'8", kind of looks tiny to me. And both my brother and I are 6'2". My dad always used to say that it was a milkman, but it just goes to show that actually, you can have two individuals with a certain phenotype, and then you can produce an offspring with a very different phenotype, right? So Mendel was really the first person that showed that, right? And that was a huge breakthrough. And amazingly, his work was just forgotten for, I think, around 50 years, right? Until we got a guy called Thomas Hunt Morgan who works on Drosophila, or worked because he's dead now, who really, he's the father of modern genetics, right? He was the one that worked out linkage, chromosome mapping, things like that. We'll be doing some of that stuff in the genetics lab in a week or two, right? So this is a pretty novel, new topic. Where we are, right? And it really covers a bunch of different things, right? In terms of Mendel's work, that's really what we mean when we say transmission genetics, right? So how do these traits or phenotypes, genotypes, right, get transmitted from one generation to the next, right? That's a case of, why do you have a particular phenotype that one of your parents has, right? And why, for a different trait, do you have a phenotype of a different parent, right? It's really a way of thinking about how those things get transmitted down generations, right? What's the probability of getting one particular phenotype over another? Pardon me. Now, obviously, that's kind of like an individual level, right? If I have a child, that child will have a mix of my wife and I traits and probably some new ones too, right? However, all of those traits really are based on the underlying molecular genetics, right? So what alleles, like versions of different genes, did my child inherit? How do those different versions of genes affect different biological processes? How do they interact, right? Does one suppress the expression of another one, right? What's the basis for those changes, right? If I have a mutation in this particular part of the gene, it'll have a very different effect if I have it in this part of the gene, right? Sorry, my hand's over here. I forgot. I have to do small gestures like this, or I put my laptop like over there at the end of my hands. One of the two. Anyway, so And really that kind of goes from, that spans a lot of different scales, right? That really goes from the like an individual bit of DNA, like its structure, how it's kind of wrapped around proteins in the nucleus, things like that, sequence of individual bases, all the way up to the control of gene expression, and even further up to genomics, right? If we want to compare, you've probably all heard this whole we have like 10% of Neanderthal DNA in our genomes, right? The way we found that out was by comparing the genomes of Neanderthals that were sequenced from, you know, bits of bones and stuff lying around to modern day humans, right? It's also how we figure out the out of Africa hypothesis, or how we support it rather. You know, how we can figure out the migration of humans. Is that me? Burr, 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 burr. Hopefully not. Oh, okay, good. Sometimes I get accused of breathing heavily into my microphone. Hopefully I'm not going to be doing that. Yeah, if you're not asking questions kind of right now, it's usually a good idea just to keep your mic muted, just in case like weird stuff happens that can bother other people. Obviously, I'll mute it anytime you have a question. Right, so how do we know that our ancient ancestors, Homo sapiens, migrated out of Africa around, what was it, 60,000 years ago, give or take? Right, how do we know that where to place like the Denisovans, right, from, I think that's Asia, compared with different kind of bones and stuff like that that we find, you know, in different kind of geological strata, right? So it kind of spans the whole gamut, right? From, you know, like studying one single gene to studying, you know, thousands and thousands of them, like comparative genomics. Or even when we're talking about, I can got cat fur everywhere. When we're talking about how likely you are to get a disease, right? So, I mean, COVID-19 is a great example. Like different people react in different ways, right? So, how do we figure out why one person gets a really bad reaction and another person never even shows any signs of symptoms, right? One of the ways that we can do that is by comparing their genomes and kind of seeing, are there different processes occurring in one person versus another, right? And then on top of that, and we're not going to look at this a huge amount, to be honest, right? Because that's what evolution is for, your the kind of sister course to this, to genetics, is if you add time, right? So evolutionary genetics is really looking at populations, typically, right? Not so much individuals and less so kind of like molecular genetics, perhaps. Looking how that stuff changes in populations over time, right? That's really what evolution is about, right? It adds the dimension of time, right? Whereas transmission genetics does have time, but it's really kind of generation to generation, right? It's fairly like a linear, small scale process. Evolutionary genetics looks at populations, like big numbers of individuals, and how those individuals and their genes that, or the alleles that they have, changes over time. And it's pretty rare to be in one camp and not another, right? So most of my, I don't do any, well, shit, actually, I used to. That's kind of funny. I used to do some evolutionary genetics, kind of accidentally. But now really most of my work is transmission and molecular genetics. Okay, what I'll also tend to do in these kind of online WebEx ones is bounce around a bit, right? So it's not as much of a linear process as the walkthroughs, right? So might get a little dizzying. I'll try and do a more or less a you know, linear fashion, but it doesn't always work out that way. Okay, so cool fun fact is that Betty Ann, you're going to need to mute your mic. That's better. Still not muted though. Betty, could you do me a favor and mute your mic? Awesome. That also tells me that she wasn't being abducted by aliens as well, which kind of sounded a little bit like. So a lot of work, like ages and ages ago, it seems at least, showed that DNA was the hereditary material, not proteins, right? Even though DNA is relatively simple, and there were 4 nucleotides: adenine, cytosine, guanine, and thymine. And also that there was, as we get to down here, we could figure out the base pairing. So we also knew that adenine was always base paired with thymine and guanine with cytosine. And the way we know that is if you kind of extract DNA from an organism and you separate, essentially the nucleotides out into piles, you'll always get 2 equal piles for A and T, and 2 equal piles, which may be different in size, for G and C, right? So if an organism, cat fur, I have to have like a little mini vac next to me next time. If you have, let's say, 30% adenine, you'll always get 30% thymine, right? Give or take a fraction percent or 2, right? And so we knew that there were 4 bases and they base paired in a very particular way, and we'll get to the mechanisms of that probably Thursday. And due to a little bit of shenanigans, Watson and Crick posited that it was a double helix, right? So there's actually someone working on, I can't remember who, thought it was a triple helix, which is pretty trippy. And obviously, that person had watched Fifth Element, which is one of my all-time favorite sci-fi films. Hopefully someone will get that reference, but if you don't, just go and watch it. Yeah. Super, super cool film. Anyway, was it Jean-Paul Gaultier who did the costume design, or Yves Saint Laurent? I can't remember which. One of the French fashion designers did the costumes for that film. Anyway, so basically they were able, with the help of Maurice Wilkins and kind of stealing the work of Rosalind Franklin, to realize that from X-ray diffraction pictures, DNA had a double helix, right? So that explains the complementarity too, right? You got 2 things, 2 strands, presumably A's, G's, C's, T's in one strand and the same in the other strand, and those pair together, right? So that kind of really brought together this model of, get out of it, DNA, right? And it's also one that I'm sure that you're very familiar with too. The one thing to point out though, which is really important for a lot of different things, DNA has direction. Actually, DNA, RNA, and proteins have direction, right? One end is different from the other end, and it goes in one particular way, right? And so in DNA, the anti-parallel, the anti part, is actually one of the most important parts. So DNA is like so, right? It's going to hurt my hand a little bit. It doesn't go like so or like so, right? It's anti-parallel, right? So one strand is in this direction, 5' to 3'. The other strand goes in the opposite direction, right? So when you actually draw out, or when I do, because I'm a little bit of a pedant, when I draw out this kind of ladder of nucleotides, technically speaking, the ones on the, oh yeah, that's a 5' end at the top, if you zoom in a little bit. The ones on the right-hand side should be upside down, right? Because they're going in the opposite direction. And that anti-parallel characteristic is super important because a bunch of different reasons, right? It's important in DNA replication. Actually causes a whole bunch of problems in DNA replication. It's really important in terms of gene direction, right? So you can actually have 2 genes going in opposite directions overlap on the same piece of DNA, right? So you can fit a lot of information into DNA because you're not just using one strand, you're using both strands. And you can do all sorts of funky, like coordinated gene expression stuff. But also that direction is translated or transcribed, sorry, into RNA, mRNA, and then that direction is what gives your polypeptide chain its direction and order, right? So essentially that one observation, that anti-parallel nature, kind of goes on into a whole bunch of other things, which we'll be learning about later, right? So that's really, really important to get. What else in here do we need to talk about? So obviously there's a bunch of stuff that comes from that, right? So all of the information that makes us who we are is encoded by, well, almost all of it is encoded by the sequences of bases, right? If we go back up here, come on, go, there we go. The sequence of bases, right? These nitrogenous bases, right? That's the information. That's our DNA code, essentially, right? And that information then gets translated into proteins, and those proteins do stuff. Now, that information is inherited over generations from one parent to another, 50-50, essentially. If you're dioecious, reproduce sexually. 100% if you reproduce asexually. Also, an important thing here is that DNA replication is semi-conservative, right? So every one of the strands of DNA that you have was originally one of the two, no, not strands, kind of every chromosome you have, one strand was from your grandparent, essentially, and one strand was newly made by your parent, right? And then you get that, and the same as it goes to every round of cell replication or cell division, right? Also, another big deal is that the only source of genetic novelty is mutations. Now, there is like one exception to all of that in terms of information and novelty, and that's something called epigenetics, right? So you can actually modify DNA and proteins for that matter, but DNA especially, so that the expression of that gene or genes or chromosome differs from individual to individual, right? Without changing the sequence of that chromosome. And those changes can be somewhat inherited, right? So we'll get to that later. And it's actually a pretty kind of crazy thing to think about. Finally, a few more words. Talk about genomics, right? So genomics is really looking at the information encoded in an organism, right, which is your genome, but looking at it on like a whole organism scale, right? We're not talking about 1 or 2 or 3 genes. We're talking about all of them, right? So we have about 20,000 genes, give or take. Worms, funnily enough, also have about 20,000 genes. Wheat has, shit, how much does wheat have? Wheat has a huge number, like 60,000 genes, right? So wheat is way more genetically advanced than we are. Yet we, spread butter on it and eat it for breakfast. But also we can do a lot of cool stuff about figuring out, how species have changed over time, essentially. And also lots of other cool, funky stuff too. Okay, so that's really the end of the intro PowerPoint. Does anybody have any questions about any of that stuff? Nope. Managed to make my coffee last until 12 o'clock, just. I didn't actually drink coffee until I was 20, largely because my parents made absolutely disgusting coffee. Now I can't imagine how I ever managed to survive without it. Okay, so again, hopefully most of that stuff is stuff you already know, right? It's really shouldn't be too much of a surprise. Can you all see me change to the second PowerPoint? Hopefully that works. Are you seeing PowerPoint number 2?
We can see it.
Okay,
I can see
cool.
it.
Yeah, these online things differ in terms of whether they share the application or share the actual individual file, like in terms of the PowerPoint. So just want to double check. Okay, tell you what, why don't we have a quick, like 5-minute break right now? All right, restroom break, go get yourself some water, something like that, because otherwise your brain's like, and my voice will run out before the end of the day. So, yeah, go get another infusion of caffeine. I'll leave you with Noelle. She can field any questions for the next few minutes. So, yeah, back in 5 minutes, say 12:15, and then we'll continue the second PowerPoint. All right, Noelle. How you doing, kitty cat? Any questions? Oh, what did I miss? Okay. So let's put a fan in. I'm not sure that's going to work very well, but we'll see. Okay. Sorry, just going to sort out stuff. Let's get that like so. All right, that's a little bit better. Time to come back, No. There she is. Noah went on a little toilet break as well, I think. Yay. Okay, so this one shouldn't take too long either. I don't want to hold you for any longer than necessary. So essentially what I want to do with this particular PowerPoint is kind of introduce kind of the sense of progression, right? Particularly in molecular genetics, right? In which we really go from, and this is kind of central dogma type stuff. So really you should already kind of have a good sense of this anyway, but there's some really important language in here that we're going to be using a lot, right? So it's going to be well worth you just kind of like genning up on this, I guess, is probably what I would say. Cat, stop moving my arm. Okay. She's very demanding. So really this is kind of reintroducing, right, this concept of the central dogma, but also the way in which it's really called gene expression, right? So a lot of people, when you say, oh, looking at gene expression today, they tend to think purely about the control of expression of transcription, right? But really, we're going to be going from like this individual structure here of a linear sequence of bases, right? And we'll get back to this in a second. And really, what we want to do is we want to end up with proteins, right? Because by and large, proteins, yes, you're very cute, are the things that do stuff, right? And that's not the only thing that does stuff. RNAs also do really cool things, but we won't go into those in huge detail right now, right? So by and large, proteins are the things we care about, right? DNA is actually, in many senses, utterly uninteresting, right? It really is purely the store of information, right? And obviously, how that information changes and how it is inherited is really interesting. But when you're looking at a particular, I don't know, genetic system or pathway, right? Sure, you're looking at the genes in that pathway, but really what you're doing when you do the experiments is you're looking at the function of the proteins in that pathway. So proteins are really the things that do stuff. Let me make that a little smaller. Also, because you are all obviously different, right? And I am different from my wife. I'm very much different from my cat. You know, how those differences occur and accumulate is also really interesting. And let's get down here just to give you like an advanced peek, essentially. And so really, mutation is where it's at, right? So there's a lot of language essentially associated with mutations, right? Because mutations have very different effects depending on where they occur and what change they induce, right? And one thing to really remember, and I stress this to every class that I teach, the mutation is random, right? It was only until very recently, CRISPR-Cas9 is probably the best example of it, that we're able to actually induce precise mutations in other organisms, maybe even including our own, if you have a distinct lack of ethics, right? So up until that point, particularly natural sources of mutation, DNA replication, or even just like various nasties like gamma radiation or UV, they produce random mutations, right? And our genome is enormous. I think something like, I don't know what the current up-to-date number is, 5% maybe, of our genome encodes for proteins, right? So huge swathes of our genome consist of stuff which is either junk, like particularly old inactive transposons, which again we'll talk about later, or stuff that we don't actually understand, right? And that's a lot of it. So where those mutations occur has a huge consequence, right? If it's in the middle of a coding region for an essential protein, say, I don't know, ribosomal subunit protein, and that occurs in your germ cells, your kids are going to be screwed, right? If it happens to occur in like some vast expanse of genomic wilderness, then most likely it'll never get expressed, right? It just very much depends. But who you are as an individual is really the sum of, what would it be now? Bit contentious, right? Potentially 4 billion years worth of mutations, right? And part of the reason, and this is getting a little bit into astrobiology, so this is how I know this stuff because I taught astrobio recently. Part of the reason why Earth is such an amazing place for life to occur is that we have an ozone layer, right? And that protects us from UV radiation, even though you can still go outside and get burnt, like in about 5 minutes if you're in Texas and you're a pasty white Brit like me. You know, I can get burned just like sun reflecting off the swimming pool into my house almost, right? But our atmosphere protects us from most of that radiation, right? So really, in terms of kind of mutation, we're in like that kind of Goldilocks zone where we have enough nasties, basically, UV and gamma radiation to induce mutation, but not so much that it completely screws us up and kills us as a life form before we have the chance to evolve, right? So if you think about, where life may or may not occur, actually how much UV that planet may receive is actually a really important part of that question, right? Because too much and you'll just damage the DNA of genetic material, right? We don't actually know what other life may use, right? Too much, and that may get damaged irrevocably, right? So that organism never gets a chance to, thrive and reproduce. Too little, and you'll never get any change, right? If you don't get change, then you don't have any material on which natural selection can act, and you don't get any kind of complex life, right? So it's kind of a cool thing to think about in terms of like, astronomical scales, essentially, right? Because we think, maybe we don't actually know for sure, that life first evolved on Earth about 4 billion years ago, right? So really, if you think about it, you as an individual are the sum of all of those changes, particularly that led down whatever evolutionary path that ended up with Homo sapiens, right? That occurred over that period of time, which is a pretty weird, that's a pretty crazy thing to think about. That's what's called giving you context, right? Probably a little bit too much context. But anyway, so one thing to bang on about again, right? And you should have got this in Gen Bio 1, but if you didn't, it doesn't hurt to go over it again, even if you did. But the directionality of DNA is really important. Obviously, these nitrogenous bases, that's what all the N's are, over here are important because that's the information, right? That's the code, those are the letters, right? Will essentially be formed into words when we get to translation. But the direction is also super important too. Leave it over here a little bit so my fan doesn't mess with my microphone. And that directionality comes from the structure of a nucleotide, right? So a nucleotide is essentially one of these, right? Hopefully you can all see me gesture kind of on this PowerPoint. But a nucleotide consists of a phosphate group, 1 if it's made into DNA or RNA, or 3 if it's just floating around on its own. Consists of a sugar moiety called this is a ribose because it's a 5-carbon sugar, right? So it's like kind of glucose, but it's only got 5 carbons, not 6, and a nitrogenous base, right? And so I always like to talk about the sugar as being like a cupcake and the oxygen being the cherry on top. So if you want to count carbons, right, you want to figure out which carbon something is attached to, you count them clockwise from that oxygen, right? So actually, can you see me using my mouse on this? Don't know if, because again, I don't know what, you
Yes.
can? Okay, cool.
Yes.
Right, so this point here, the carbons themselves aren't directly labeled, they're just kind of implied by these points. That's the first carbon, right? So the nitrogenous base is always attached to the first carbon. And also, it's kind of neat in this picture, you'll also see like an amine group here, 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 an oxygen here. Those are what's involved in hydrogen bonding, right? Those are the bonds that hold those two strands together, right? And typically what you'll find is that the number of hydrogen bonds is determined by how many places those bonds can form, right? And I'm not an expert at this, but for example, you'll get a hydrogen bond between an amine group and say an oxygen group on the other side. Anyway, so if we keep counting round, this is DNA. Here you go. Here's a question. Other than the fact that it says it in the title of the slide, how do we know that this is DNA and not RNA? Question to the group. See if anybody can answer that. You can answer either like speaking it out loud or typing it up. It's up to you. What are the two differences between DNA and RNA, generally speaking?
Is it the way that these strands are facing?
Who was that?
Gabrielle.
Oh, hey, Gabrielle. Sorry, it doesn't always come up with who's talking. Not necessarily. No, DNA and RNA are both directional and they can both base pair.
Doesn't RNA change a base pair?
It does. Can you remember which one?
Is it T?
Yeah, T gets changed to a uracil, so that's one change. But we don't have either a thymine or a uracil here, so there's another change or difference, which is really important.
Mm,
And don't worry
not
if you
sure.
don't know it, because that's kind of why I'm here to tell you, right? So you see here on this second carbon, there's a hydrogen. So in RNA or an RNA nucleotide, there'll be another hydroxyl group here, right? So that's why RNA is ribonucleic acid, right? Whereas DNA is deoxyribonucleic acid. It's deoxy because it has a hydroxyl group removed from it. That's where the deoxy part comes from. That's one of the main differences between DNA and RNA. Anyway, that was just an aside. So the third carbon, though, is one of the important ones, right? So the third carbon, which you see down here, pardon me, is where you have a hydroxyl group. Now, can anybody remember the name of the reaction? There's a couple of different names for it that joins nucleotides together. Same for proteins, for sugars. Anybody remember what that's called? Produces water.
What does it start with? What's the letter?
Either a D or a C. It also happens a lot. It doesn't happen on this because this is a double-walled thing, which is kind of handy. But if you have a glass full of cold water in Texas, particularly San Antonio, what are you going to get on the outside of it and then puddling around the bottom?
Precipitation, isn't it?
Condensation.
Condensation. Yeah, precipitation would be a little scary.
Hydration, dehydration, that's right.
That's the one. So it's a dehydration or condensation reaction. The same, different words for the same thing, right? And so that hydroxyl group here is really important for that condensation reaction to occur because it either provides a hydrogen or a hydrogen and oxygen to make water with, right? And so that 3' OH is the only place where another nucleotide can be added, right? So when you're synthesizing DNA or RNA for that matter, right, that's the same principle, you can only add a new nucleotide on the end here. You can't add it on at the 5' end, right? So you can only make DNA in one direction. And because you can only make RNA in one direction, right, for the same reasons, you're going to read DNA in one direction and make RNA in the same direction, right? So that's where the transcribing comes from in transcription, right? So these are like kind of small facts which on their own may seem inconsequential, right? They just don't really matter much, but really they underpin like all of, well, I mean, really all of biology, right? Because all biology is based on either DNA or RNA. And so you keep counting around, there's a 4' carbon here. Get off it. I'm not getting my pointer anymore, right? In that corner, and then the one above it is the 5' carbon. And so that is the one which has the phosphate group attached, right? So if you were to look at this diagram here, essentially this part in the middle, right, from the phosphate down to that O, right, attached to that sugar, that is one nucleotide, right? Here's another one down here. And if you had a free one, which you wanted to add on, it would come in at the bottom, it would lose a couple of the phosphate groups, and then you get this phosphodiester bond formed, right, which is part of that. Essentially, it's what makes the backbone of DNA. And that's a real common thing in kind of macromolecules, particularly nucleotides. Yeah, nucleic acids, fats, to some extent, and proteins have a common backbone, right? So this here is the phosphate sugar backbone because it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on, right, forever and ever, right? That's the backbone. That's the same whatever your DNA sequence, right? And then you have the individual differences kind of hanging off of that. That's exactly the same for proteins as well. Anyway, so that's what the difference is all about, or the direction rather, right? So DNA is always from 5' to 3' end. It's made in that direction, and it's read, as in made into mRNA in that direction too. Right, I'm not going to talk about this much because this is really a copy of the slide you've seen already. Okay, so gene expression is really the term. Oh, cool. We've got lots of answers in the chat section. Yeah, you're all right, all of you as well. Sorry, I didn't see that until it was too late. In terms of gene expression, we're talking about going from DNA, which is the storage molecule, right? That's what stores the information, to protein, with mRNA as like an intermediate step, right? And that whole process is gene expression, right? So the information that's encoded by DNA is obviously important, but it's not what does anything. It's not what's actually expressed in terms of phenotype. That's really based on what protein is produced, how well it functions, and what effect any changes in that protein or its expression have on a particular trait. And so that's best summed up here. I actually hate using this. It's not the central dogma, it's the sequence hypothesis. But again, I'm a little bit of a pedant when it comes to stuff like that. But really what this is showing is this kind of linear flow of information, right? From DNA to protein. DNA stays in the nucleus, right? You don't take it out of the nucleus and well, I mean, you'll only see that happen if something catastrophic happens. And so essentially you need an intermediate, right? That's one reason for having mRNA. The other one is that you can control how long that mRNA hangs around for as well, right? So rather than just essentially turning on or off gene expression or the transcription of that particular gene, you can also kind of clear up the mRNA afterwards as well, which is another way to control how much protein you produce. So transcription is really the process of producing RNA, and there are lots and lots of different types of RNA. 3 here, only one of them is actually directly translated into protein, and that's messenger RNA. Ribosomal RNA is the enzyme. It's called a ribozyme, which actually catalyzes peptide bond formation. And tRNA is really the kind of the thing that translates nucleic acid into amino acid, right? So this is the intermediate between mRNA and protein. And then all of that together, that's called translation, basically. Right, so it pays to remember this kind of flow, right? And there are lots of different ways, and we'll get to the in the good of time, how you can control each individual step, right? Actually, like, it's called gene regulation. It's a crazy field. It's astonishingly complex, right? There are so very many ways in which you control it, a few of which we'll be talking about. Another key point to make, right? So here we go. This is the RNA transcript down here. And now you can actually see that uracil come into play, right? So here's our RNA strand. So this is it's actually kind of hard. I need to bring up my kind of board. We'll cover it again in a little bit anyway. But in DNA, you have the strand which has the information, right, that you're copying, and then you have a template strand, right? The mRNA is actually being made using the template strand, but in doing so, it will have the same sequence, with uracils obviously going in instead, as the actual gene itself, right? So we'll clarify that again later, because that's a little bit kind of trippy to think about. So basically, even though we have 2 strands, only one of them is being used for copying that DNA into mRNA. Also, when we talk about genes, there's really 2 key parts to a gene. One is the bit that's copied into mRNA, right, which is what you can see going on here. But there's also another part, which can be huge, which controls when and where and how much that happens, right? So it's not enough just to have the information. You also have to have the ability to control how that information is accessed. right? And that's a very big part of gene expression. This stuff you already know. Okay, this is another thing which is kind of worth kind of thinking about a bit, right? So essentially, let's go back here. What this is, and this mRNA transcript will be as well, is a series of letters, right? You've got A, C's, G's, and U's in mRNA at least. But you only have 4 of them, right? And so if one letter equals 1 amino acid, you get 4 amino acids, right? So there are more than that. There are 20 different amino acids. And the only way you can get 20 different amino acids from 4 nucleotides, from 4 bases, pardon me, is if you bundle those letters into words. So all of these letters, they're going to get put into 3-letter words. Those are called codons, right? And each one of those 3-letter words equals 1 amino acid, right? This is a really important point to get. Now there can be different words that equal the same amino acid. That's called degeneracy, right? But one codon can only ever equal 1 amino acid, right? And so this is really what you could call like the Rosetta Stone, right? So the Rosetta Stone, a little bit of history for you. People couldn't read hieroglyphics, right? Because as a language, you know, you got like people wandering around with like birds and, you know, stuff like that. No one knew what it was all about, right? So no one could actually read hieroglyphics like from the ancient Egyptians. All those inscriptions in like the pyramids and tablets and things like that, didn't know what it meant. And yet, so what we needed was like some kind of intermediary, right? We needed like a decoder, essentially. And that was found in Rosetta. It's called the Rosetta Stone. It basically had some hieroglyphics and some ancient something or other, Sumerian or Greek or who knows what, on it. And so we're able, or Egyptologists were, to be able to start decoding hieroglyphics. And that's really what this is, right? So this is essentially our way of being able to say, okay, for any given word, right, our 3-letter word in RNA, we can figure out what amino acid we're going to get, right? And so you'll find that there are 2 amino acids which are encoded only by one word, one codon. That's tryptophan and methionine, right? Methionine is an important one, which we'll get to in a little bit. But there are others which could have up to 6 different words, still give the same one. I think leucine is one of those, right? Isoleucine only has 3. They kind of, you know, jumped around a little bit. So there's a lot of redundancy going on here, and that's actually pretty good in terms of robustness, right? If you've got a really important gene and you get mutations that change one of these bases, then you stand a good chance of actually ending up with the same amino acid, right? So it's a kind of a buffer. Another important point to note is that there are 2 lots of kind of very specific codons here. We've got a start codon, that's our methionine, right? That is always the first codon, always in translation, right? And we'll look at that later on, kind of to kind of put that into more context. And there are 3 codons which don't code for any amino acid. Those are called termination codons or stop codons, right? And these don't have any tRNA coming in to recognize them, right? So as soon as one of those gets hit, the whole ribosome kind of trundles along, hits one of those and just stops. Then something comes in and just flicks it off, right? So that's what stops translation, like the whole process, right? So you have a very discrete start with the methionine, the start codon, and a very definitive stop, right? So you'll only get protein produced from the sequence or translated from the sequence between those 2 things. And that decoding, that translation, essentially occurs using tRNAs, right? And these tRNAs are kind of, shown here. You've got an even fancier one here, right? So this is a, oh, actually, no, I'm sorry. That's ribosomal RNA. Scratch that. This is a tRNA. So tRNAs are what allow that translation to occur. Basically, what they do is they have part of them that recognizes that word, and one tRNA will only ever recognize one word, right? It will never go in and kind of match up with another word. It just doesn't work, right? So they have a very specific sequence called an anticodon, which recognizes a specific codon. And that anticodon is also used to add on the correct amino acid, right? So that codon here in the mRNA is recognized by the tRNA anticodon, and that brings with it the correct amino acid for that codon, right? So that's essentially how the mRNA is translated into protein, and that happens in a step-by-step process, essentially. Another quick point to make, which I've already alluded to before, RNAs are very varied, right? There are a huge number. There's at least 20 different RNAs last time I looked, right? Different types of RNAs. We've obviously got mRNA, we've got ribosomal RNA, tRNA, which is what these things are, transfer RNA. But we also have small interferon RNAs, microRNAs, there's a whole gamut of them. And they can form real funky structures too. So they can actually fold, and they can base pair with themselves. They can form kind of more complex structures, right? And those structures allow them to do stuff. So here's another little fun aside. RNA is the only thing which is both information and an enzyme. Not all RNAs, but some of them. So RNA, there's actually a thing called the RNA world, where a lot of scientists that work on this stuff think that RNA was the first form of genetic information, right, in terms of the evolution of life, right? Because DNA can't do jack. It's just sitting there. It just gets turned into something useful, right? RNA is the only thing that can both store information and catalyze reactions, right, as an enzyme. And so in theory, an RNA could copy itself, right? And so then you get the basis for transmission of information from one generation to the next, and potentially evolution. Okay, so we're going to wrap up with a quick look at mutations. Again, mutations are only source of genetic novelty, and generally speaking, they're bad for you, right? They're either bad or indifferent, right? It's very rare that a mutation will increase the fitness of either a gene or an individual. Usually they'll cause problems or make no difference whatsoever, right? And so mostly, generally speaking, the vast majority of human diseases, other than ones which depend heavily on environmental interactions, right? Most of them are due to mutations in particular genes and particular processes, right? So sickle cell anemia is a mutation in hemoglobin structure, causes, instead of nice like donut type red blood cells, you get like these horrible kind of curved sickle-shaped cells, which are really painful, don't actually store much oxygen. A lot of cancers are caused by mutations either in genes which control cell division or prevent it, right? And so on and so forth. Down syndrome isn't actually technically a mutation. I should take this out of here, to be honest. It's due to an extra chromosome, right? Which again could be due to mutations elsewhere in DNA replication and cell division and DNA replication. And so really when you think about it, and this is a very natty hand-drawn picture, really mutation is the source of novelty, right? But it depends on what effect that has whether or not that change is ever going to get propagated, right? So if you think about it, right, you're an individual, mutation occurs during cell division in you. That mutation, you got one version of it, one copy in a population, right? So if you're actually going to see that variation, kind of a higher percentage, you've got to have some way of selecting for it, right? And so that's really what comes in with environmental pressure and natural selection, right? So a mutation in a cell surface receptor, for example, called CCR5, confers resistance to smallpox. Also confers resistance to HIV as well, right? And so back in the day, which wasn't actually that long ago, smallpox killed, they actually think, what was the numbers? I have to remember. I think about 1.5 billion people have been killed by smallpox over the course of human history, right? So that is what we call an environmental pressure, right? And so anything that protects you from smallpox, that change is going to spread through our population, right? Particularly if it allows those individuals to reproduce and then pass it on to their offspring. And so over time, the frequency of that change, that allele will increase, improve that trait, fitness of the population will improve, and potentially over enough time, you'll end up with differences between populations which result in speciation, right? So again, it's very much a matter of time. And they think for that particular mutation, they think it took about 700 years for the frequency of that change to go to about, I think it's about 10% of Northern European population have that allele, right? So that just kind of gives you an idea, and that's an enormous selection pressure. Typically, things are going to take more likely in the tens of thousands or hundreds of thousands of years to change in frequency. And also, I'm not going to go through this in any particular detail because it's, it's a table and you can do that yourself. But the consequences of that mutation vary enormously depending on where it is, right? In terms of a gene, is it in a gene itself? Is it in just DNA outside of a gene? Is it affecting regulation? Is it affecting function? What kind of change is it? And does that affect the polypeptide sequence of a protein? Because you can have a mutation, which in this case it's called a synonymous or silent mutation, has no effect on protein sequence whatsoever. Because if we go up here, you can see why, right? So if you have a codon for phenylalanine, 3 U's in a row, and for whatever reason that third U gets changed to a cytosine, no change. You'll still get a phenylalanine, right? So because of this degeneracy, right, a lot of mutations, even if they occur in the protein coding region of a gene, are actually silent. Now, you can also get one which causes a frame shift, and then you get a stop codon pop up. That's terrible. It completely gets rid of that protein function totally, right? But again, depends what kind of protein that is. If it's something you need to survive, then that's not good. If it's something that, just makes you a little bit sexier to the opposite sex, maybe that's a good thing, maybe it's not a good thing, but it's not going to necessarily change your fitness in a huge way, right? So there's a bunch of different terms in here which we'll be coming back to later on in the course, which are useful to know. So it's a good idea to just kind of be familiar with some of them. And to be honest, that's pretty much it for me for today in terms of lecturing. Again, most of this is about terminology, language, but also giving you the general overview concept of what gene expression is all about, because we'll be going into each of these steps in pretty good, high detail essentially later on. So it's good to get this as a general concept so that when we get into the weeds, you're not going to get too lost, right? It's kind of an important part of, succeeding in this course, essentially. Okay, anybody got any questions? You're probably all thinking, I want to have my lunch, right? But just in case you can, hold off eating for a little bit, if you got any questions, I am all ears. Again, if you don't, that's also cool. And if they come to you afterwards, you can always just bring these to next lecture on Thursday too, right? So I've got absolutely no issue with going back over previous PowerPoints. Typically, we're going to have plenty enough time for covering all the material and then some. So anyway, if you have questions, and ask them now. If you don't want to ask them now, you can ask them by email or ask them in class on Thursday. And if not, then I'll see you again in, 50 minutes? 1:45 for lab. That's going to be really weird teaching a lab online, but hopefully we'll make it work somehow or other. All right, any questions? Last call? No? Awesome. Okay, so I'm going to stop the recording now. I will post up. I'll actually see what the chat looks like. If there's any private chats, I'll see about taking those out because people may not. Hey, stop chewing that. Sorry, she's nibbling on my headset cable. If there are any private chats that people might not want included, I'll take those out. But I'll post up this recording and the chat record of the chats to Blackboard on YouTube, right? But as soon as I do that, I'll let you know, and I'll send you the links as well. All right.
Thank you so much.
Oh, someone said something.
Thank you.
You're welcome. Cool beans. All right. See you in about 50 minutes.